Hello everyone and welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping housed in the Martinos Center. It is our great pleasure to introduce our speaker today, Dr. James Morris. James Morris is professor and chair of the Department of Psychology at the University of Virginia. He earned his PhD in biopsychology at Stony Brook University in 2003. After earning his degree, Morris completed postdoctoral training in functional neuroimaging at the Brain Imaging and Analysis Center at Duke University, where he was supervised by Gregory McAfee. While at Duke, he began collaborating with Kevin Flavery on studies investigating typical and atypical social perceptual processes in children with and without autism spectrum disorders. In 2009, Professor Morris joined the psychology department at the University of Virginia, where he directed the University of Virginia Social Neuroscience Laboratory. Work in this laboratory focuses on individual variability in the social functional across the lifespan, with a recent emphasis on social and emotional development in preschool children. Along with his collaborations, Professor Morris employs a wide range of techniques, including structural and functional neuroimaging, epigenetic assays, EEG and ERPS, and eye trackings, and behavior and data science approaches. I would like to remind our audience to please address any questions using Q&A box or raise their hands at the end of the talk. Dr. Morris, thank you very much for coming today, and the stage is yours.
Thank you. Thanks for inviting me. Looking forward to sharing some results from the lab from the past couple of years and one of our particular areas of focus. As stated, I am a professor at the University of Virginia in the Department of Psychology. I've been doing functional neuroimaging work since about 2003. After I got my PhD, during my postdoc, I was trained to do functional neuroimaging work and got my start really looking at social cognition, social perception, and the neural system supporting these behaviors in both typically developing and individuals with autism. And most of my work at UVA has focused on adults, particularly pre-tenure. I spent a lot of time focusing on individuals 18 to 30. Since that time, because of some of the nature of our research, we've pivoted to take more of a lifespan approach, and this has led us to test infants as young as a couple months old to the elderly all the way up to 80 years old. So we do have some sort of lines in those programs as well. What I want to talk to you today about is work that we've been doing on a longitudinal sample of kids that we started with some pilot money several years ago. And this has kind of grown into the point where we started small with what we basically could afford from an internal grant. And we're hoping to continue to build because we think some of the preliminary findings are really fascinating, and they're well supported by hypotheses we might derive from the animal model literature, as well as from some of the functional literature that's been here before. So I'm going to talk a little bit about that today and hopefully get some feedback on that. Okay, so just to give you some central hypotheses to orient you where we're going. I'll first mention my two collaborators on this project. This project actually has multiple collaborators, but central to this project are my longtime collaborator, Jess Connolly, who's a professor in the neuroscience area of the psychology department here at UVA, and a graduate student that we shared named Amalia Skyberg. Amalia graduated with her PhD last year. She's now a postdoc at the University of Oregon. Okay, so in this study, we really started with these central hypotheses that were based on a lot of the work coming out of Connolly's lab and some of the work that came out of our imaging projects together. One central hypothesis, or a couple central hypotheses that we base sort of this work on, starts with one, that early social experience can epigenetically modify the endogenous oxytocin system. I'll briefly touch on what I mean by this. We have some strong evidence of this in the model system prairie vole, And we have some evidence of this in human infants, and we see some of this in our preschool kids as well. We have a nice background on this now and have some ideas about how this is working. We then hypothesized that these modifications shape neural developmental trajectories. So just as the environment can influence the genome, we also suspect that it influences neural development, in particular pace of development, and perhaps shaping brains that are more capable of handling self-regulatory processes. And I'll talk about what I mean by that as well. And then finally, this creates trajectory differences in social and cognitive capabilities and behavioral outcomes, things that we might be interested in as kids enter school age. So we use terms such as school readiness to basically define this idea that a kid is ready to be able to learn from a cognitive standpoint. They're able to emotionally regulate themselves around others in classrooms, and they're able to make friends and to engage in social activities that add to their developmental experience as well. And so I'll talk a little bit about how we conceptualize these things, measure them, and how they fit into our hypotheses and how our data supports these ideas. So years ago now, Connolly, working with her postdoc advisor, Simon Gregory at Duke, were assessing copy number variant differences in autism. And they were specifically interested in looking at post-mortem tissue that was donated from autistic brains, as well as typicals for comparison purposes. And during this time, they also looked into this area along the promoter of the oxytocin receptor, and noticed that there was higher levels of DNA methylation along the promoter region at these specific sites from this brain tissue in individuals with autism relative to control participant brains. And what was interesting about it is that it sort of followed this canonical pattern that we often talk about when we talk about DNA methylation, which is that there's more methylation leads to reduced gene expression. So it compacts the chromatin and does not allow for as much gene expression. And therefore, you would presume that there'd be less oxytocin receptor. This story gets much more complicated in the animal model systems and work that's now come since that time. But for an easy sort of toy takeaway in this case, we think of higher levels of methylation equals lower levels of gene expression. This is a way that we can epigenetically govern the oxytocin system itself. And so you can see here, this is from brain tissue. Epigenetic processes are tissue specific. So one major issue here is how can we assess this in a living cohort of people? Well, since that time, we've established that there is this relationship between OXTR level and expression in the brain tissue. They also, in the original paper, were able to demonstrate that this pattern persists in the blood as well. So accessible peripheral tissue, in this case, mononuclear cells. You can see there's a difference between individuals with control with and without autism here in the blood as well. And since that time, we've also correlated blood and saliva levels of this measurement to see if we could easily assess this from saliva. And this is obviously important when you get into studies of young children. Not many people are willing to volunteer their kids for studies doing blood draws. All our original work in the imaging field with OXTR methylation has been done on blood. We, since that time, have gone back and established that we can do it with saliva as well. And this allows us to get into these pediatric populations without causing undue stress. So just to give you a little bit more background, as I mentioned it before, a lot of our work is informed by the prairie vole. Some of you might have heard of the prairie vole before when talking about pair bonding type behaviors and social type behaviors. It's an interesting rodent species in that it is considered to be a monogamous species, is considered to be a species that pair bonds with another and shares to some degree in the parenting and the care for young pups. And so these type of social behaviors are shared with mammals like humans. And they make them interesting species to study when you're studying such things as bonds and formation, pair bonding formation, and other sort of complex social behaviors. Now, natural variation in parenting is a newer phenomenon in prairie voles. A lot of times, a lot of the early studies look at things such as neglect or taking a pup away from the parents, and then assessing the impact on various systems. But there's also a fair degree of natural variation in parenting. And natural variation in parenting would just simply refer to some parents do more sort of pro-social types of behaviors, more sort of affiliative bonding types of behaviors, such as licking and grooming and other things that we can easily measure and assess in the lab. And we know that these behaviors matter in terms of development. So natural variation in parenting is associated with the rate of maturation in these voles. So you can see those with high contact parents, the ones that do more of the huddling, the licking, grooming, more high care parenting, tend to develop slower than those with low contact. So in this case, you see, the age at leaving the nest, for instance, is postpartum day or post birthday 14 in this case, and those with low contact leave the nest sooner than that, closer to day 12. So development happens quick in this species, but this is just to point out that this ability, this amount of parental care actually does change the impact and the trajectory of development, and presumably through neural development as well. So the model that we work with this species is, I don't know why this isn't going forward. There we go. Okay, so the model that we work with in this species looks something like this, where we have decreased parental care is associated with decreased social behaviors and faster development. So we also see behaviorally that these animals don't engage themselves in as many social behaviors. Ones that had more low contact parents are more low contact themselves. They have increased OXTR methylation. So the pups from low care parents have higher levels of oxytocin receptor methylation. They have decreased oxytocin receptor gene expression, decreased oxytocin receptor binding, which you can see in these little slides here. And this is going to lead to this trajectory where, because of this cascade of influence between behavior and manipulation of the genome in a critical system related to pair bonding, it's going to influence social behaviors throughout the lifespan. And this is what Connolly and colleagues have been able to establish in the prairie vole system. And we're interested in how much of this impacts humans. Humans also, we have a lot of varying degrees of natural variation in parenting, even in really good situations. And then obviously we also have really bad situations where we have early neglect or unfavorable, impoverished environments, which can impact the social environment in which a kid grows up in. Okay, so here's a figure from some of our human work, and this comes from Katie Kroll and colleagues. And essentially what they had done is they looked at oxytocin receptor methylation at 5 months, and then they looked at it again a year later, a little over a year later. And what they had found is that in general, the oxytocin receptor methylation in the mothers doesn't change. It seems to be somewhat stable. You can see that in the red figure there. There's a little bit of change. Some of the changes is error variance that comes from the assay. Some of the change is real, but it's in general pretty stable. Whereas the kids, you end up seeing there's much more dynamic change in oxytocin receptor methylation across this time. And when you look at maternal engagement at 5 months, so when you look at measuring maternal engagement, and ways that we might do this is through some natural viewing. We might create some paradigms. We might have some observational sort of ways at which we assess the amount that the mother engages with the child. And then we measure oxytocin receptor methylation. The degree of maternal engagement predicts the degree of oxytocin receptor methylation, where those that are higher in maternal engagement have, those infants have lower levels of oxytocin receptor methylation. So this is very similar to what we find in our prairie vole model. So just to give you this background, to give you the summary where we're going to launch from, in humans, oxytocin receptor methylation is dynamic in infancy, and it can be predicted by the degree of maternal care, maternal engagement. In prairie voles, early care, the amount of engagement from the parents can alter the oxytocin receptor methylation state, which then modulates later social behavior. We can capture this across the lifespan easier because, as I said, development goes quick in the vole. Their lifespans are shorter. We can actually assess them later themselves as parents and assess their social behaviors and how this lasts across the lifespan. Okay, so stepping out of that world and now going back to the world of imaging and thinking about what we know about social emotional behaviors during development. One of the findings that gets reported often in the field of neuroimaging, cognitive neuroscience, developmental cognitive neuroscience is regards the connectivity between prefrontal areas and the limbic system, specifically the amygdala. Emotional regulation follows this predictable developmental course. So when children are really young, when they're infants all the way until they start getting into preschool, they're not good at regulating emotion. Anyone with young children will know this. They're not very good at self-regulation, and they often require caregivers to help them with that. It's a difficult environment. It's one with lots of changes. There are lots of factors that go into play, and parents oftentimes will help children navigate and keep them under control, soothe them when things go wrong. But across the lifespan, as the child goes from those very early days through adolescence, they acquire more abilities to do this. In the state of Virginia, the way we assess school readiness, emotion regulation is one of the foundational points of this. How well is a kid equipped to regulate their emotions when they reach kindergarten age, when they're ready for school age? As I said, bidirectional connections between the amygdala and prefrontal cortex are well characterized in emotion regulation. And it goes kind of like this. This is a figure I borrowed from Tottenham's group. But the idea is that really early in childhood, you see this positive directionality in terms of connectivity between the prefrontal cortex and the amygdala. So when a child is viewing provocative image, some angry faces or other sorts of emotionally evoking type of stimulus, you'll actually see that both areas are ramped up in response to the stimulus. So it's a sort of a circuit of arousal in this case, one that is really sort of indicating the valence of the situation, and both amygdala and prefrontal cortex are showing stronger positive response in this case. As you get later in childhood and into adolescence, you start to see a shift in the profile of connectivity here, such that when you have larger sort of PFC activity, you have less reduced amygdala activity. And this has led many people to speculate that this circuit is tremendously important for things such as mood disorders, self-regulatory capacity, emotion regulation, many of the things that we find foundational for social and emotional behaviors. So adulthood is characterized by this negative relationship between these areas, negative functional connectivity between these areas. So you find a developmental shift in this way. So here's another sort of way of looking at this. The amygdala reactivity to fearful faces decreases with age, and this is likely due to top-down regulation. So the amygdala itself, the activity in the amygdala decreases across age, and this is presumed to indicate more self-regulatory processes that help tamp down the processes here. You can see this in this figure by Dylan Gee from Journal of Neuroscience 2013, children 4 to 9 with this really strong connectivity between amygdala and prefrontal cortex areas. So this is a positive profile. Across the lifespan, this becomes a negative profile. So where, as I mentioned before, this becomes negatively correlated. Okay. Also in this paper, this is just an impact of early environment and why we think this matters, and gives us some sort of tie into our own work in this case. You can see from this paper where they not only tested children from a sample where you might collect here in Charlottesville or somewhere else, a sample typically raised children show this pattern where you have this positive connectivity and this negative connectivity as they get into adolescence. You can see this in a second sample as well here. And then they also looked at children that were institutionalized early in life. So these were orphans, Romanian orphans that had, basically been living in institutions since birth. And what they have is they have sort of very early sort of indicators that their connectivity between amygdala and prefrontal cortex is negative. It looks more like the adolescent or adult-like profile. This could be thought of to be somewhat of a compensatory mechanism. So in a environment that's largely doesn't have as much connection, doesn't have as much interaction, doesn't have as much proximity with caretakers and the child, their neural systems are developing quicker. This circuit is developing quicker. More recent work has assessed a number of biological measures in kids that grow up in an unfavorable environment. And you often hear stories of early maturation and different marks of early maturation in these cases. So it's assumed to go along with this as well. So a working hypothesis of ours is that middle childhood is a sensitive time in the development in which oxytocin receptor methylation may serve as an adaptive mechanism in the rate of socio-emotional development. So here, kids that are in an unfavorable environment might have increased OXTR methylation. So this would match up with what we saw with some of our infant studies and with the vole studies. They have quicker development. We should see faster maturation of neural systems supporting social emotional behaviors. And those from more favorable environments will have less oxytocin receptor methylation, and they'll have a longer, prolonged sort of maturation, including maturation of neural systems that's longer, which leads to different outcomes from a social emotional standpoint. And I'll get into what I think would be really interesting questions that we don't have the answers to on this side as well, because I think that this really raises questions about what is good development and what isn't. Okay, so as I said, several years ago, with a pilot grant, we started testing kids, and we started following a longitudinal sample. We recruited kids from our local community. So at the beginning of the study, this isn't the most diverse group of kids because we were largely counting on people we could recruit easily. There could, if we want to be scientific, it'd be better to have more variability in terms of early social experience environment. It's a limitation of our study that we don't have that. I will get to one caveat about that later, which is that we did follow these kids before and then during and after COVID, which did introduce a fair amount of stress in an otherwise sort of, really secure environment. And we'll talk about some of that implications as well. So the first parts of the study, we really tested a wide range of kids. And we worked to get our methods under control here. So, two big issues here were, one was to figure out if we could use saliva to assay oxytocin receptor methylation, which I pointed out before we can. And the other was can we get our kids to stay still enough to make these connectivity studies accessible? And in both cases, I think we were successful. We had a rigorous sort of mock scanning routine that we did before we put the kids in the scanner. For resting state scans, we used these Inscape movies, which are these movies that just have light sound and they have some moving geometric shapes, but no real content. They're a little bit more soothing, a little better than staring at a fixation cross. So we use these with our kids based off of some of the prior literature that's used these to reduce head motion. You can see across our paradigms, this is just a regular functional paradigm where we do a block design of animate versus inanimate objects. We do a lot of free viewing with movies. In our case, the kids watch clips from the movie Finding Nemo, Pixar movie, which has a lot of interaction and drives up a lot of systems. And then we use this Inscapes for resting state scans. We also collect diffusion tensor imaging, and we use diffusion tensor and diffusion kurtosis to analyze white matter microstructure and tractography in these participants, and I'll report on that a little bit today too. Okay, so just to get to the basics, we have a lot of adult data laying around, so we've taken some of our adult data and we looked at the differences in connectivity just to establish what we have found in prior literature and what's been reported. And you can see the pattern does persist here in our own data set, where children have this more immature profile where there's a stronger amount of connectivity between amygdala and prefrontal cortex in this case. So more childlike would be up here and more adult-like would be down here. And adults are significantly different in that they have this more neutral or negative differences between connectivity between these regions. I should note here that we didn't really drive the system in this case. We didn't use the emotional faces. This is basically coming from rest while they're watching inscapes. So there are some differences between some of the reported activation that you might have seen in some previous literature, but consistent with the overall pattern that one would suspect or hypothesize when looking at adults versus kids. In terms of our targeted hypothesis, oxytocin receptor methylation is negatively associated with this amygdala medial prefrontal cortex functional connectivity. So this is controlling for age. So this isn't using chronological age. We did have a larger range of age in our longitudinal sample. So age is a consideration when thinking about things that could be driving this difference, given that this is a developmental process. But when controlling for age, you still find this difference whereby the kids that have more oxytocin receptor methylation have this more adult-like profile of functional connectivity. So this more negative to neutral difference or connectivity between medial prefrontal cortex and amygdala. Okay, we also find that self-control is negatively associated with amygdala and MPFC functional connectivity. So in this case, we used an assessment of parent-reported self-control, and we find that parents that report their kids as having more self-control have these children are ones with more adult-like profiles in this case. So this is, right in line with what we would predict. Kids are developing this circuit, which is helping with these self-regulatory processes, which is allowing kids to keep themselves more under control in emotionally challenging situations. My child remains calm when teased is one of the assessment sort of items on the self-control assessment in this case. Okay, we also have white matter tritography differences. So here we quantified the streamlines. We quantified the number of streamlines between amygdala and MPFC from our two targeted areas. We functionally define the areas, and then we assess them with our DKI analysis. This was work done by Ben Newman, who now works at the School of Data Sciences at UVA. Ben specifically is an expert in the application of 3-tissue composition of white matter data, white matter microstructure, and he looks at a lot of different patient populations as well as developmental populations. And he helped us with this analysis here. And what we find is that there's structural evidence to support this hypothesized top-down regulation of amygdala reactivity and mature connectivity networks, such that those with these higher levels of OXTR methylation have more streamlines, they have more development that's taken place between these areas of white matter tritography, which perhaps is supporting these self-regulatory processes. Finally, as I mentioned before, we collected all this data, and then 2020 rolled around and COVID sent everyone home. We didn't collect kids' data, functional MRI data for a couple of years during that period of time due to restrictions placed on us and also for safety precautions and things. But we did, as I said, have a community sample where we could test them on other things. And we had come to use some of that time to drive iPads over to the houses of families and leave them with the kids to engage in a number of different assessments. And one of the things that we had found was that, everyone to some degree, probably as a parent myself, I can speak to this, had some level of increased stress during that time. The uncertainty of school schedules, the uncertainty of daily schedules, financial, any kind of number of factors could have influenced it. So in general, to what we see typically reported by parents, there was more stress in these households and such. So it did drive what was a fairly not representative sample of the US population to a little bit of a different place. And so a little bit more, stress than we would typically see. So what we had asked is whether or not this relationship between oxytocin receptor methylation and this amygdala MPFC functional connectivity is predictive of internalizing behaviors 2 years later. So 2 years later, we measured internalizing behaviors. And these are the types of things that people often are worried about when it comes to early signs for increased potential for mood disorders, psychiatric disorders, anxiety, depression, these types of things where you're engaged in such habits as I'm blaming myself for these outcomes, or being overly hard on myself in this situation, or no one likes me, or things like that. And this is different than externalizing behaviors, which are more measurable outbursts and sometimes violent activities and things like that. Well, what we find here is this interesting pattern that I'll try to unpack here. These individuals that are high in OXTR receptor methylation and have this more adult-like profile are buffered somewhat from internalizing behavior. They scored low on internalizing behaviors 2 years later. So the children that are maturing quicker, that have this higher level of oxytocin receptor methylation, they're showing this buffering effect where they're more protective from internalizing behavior 2 years later. The kids that are low on OXTR methylation, they just generally tend to be more of a flat line here. Not many of them actually score very high in terms of internalizing behavior. You have this one sort of outlier here that does, but in general, we don't see as much of that. And you also see a slower rate of development in these kids. So these kids seem to be not yet reaching that peak where internalizing behaviors is becoming as big of an issue or on the radar as much. And they're also not developed as much as the kids that have the higher levels of oxytocin receptor methylation. And so I'll get back to that in a little bit. And this is just showing you some of the statistics behind it, showing this Bayesian analysis here to demonstrate that this is a real fact. So a more adult-like amygdala MPFC connectivity is associated with higher levels of oxytocin receptor methylation and greater self-control. And this accelerated connectivity profile might actually be suggestive of an adaptive buffering to internalizing symptoms. So the brain might be showing some level of earlier maturation of these systems, which is then having a behavioral outcome. And for someone that does a lot of imaging work, this is a really neat finding because it not only has this association between what would be a really plausible biological system under epigenetic control, but we also can see a behavioral outcome in this case, which is not always easy to find. Okay, so the other part of our study, and one of the things that I wanted to talk about because it kind of talks about the other side of this, is that we not only have resting state data, but we've also used data that comes with free viewing of complex situations. So in our resting data, we have inscapes, as I said, which is kind of like the 90s screensaver-ish, moving shapes and such, but there's no content to process. It doesn't necessarily drive a lot of systems involved with understanding content, emotional systems, attentional systems, things like that. Whereas movie watching can really get all this in one fell swoop, and we don't have to worry about certain performance metrics that come along with complicated functional tasks. So what we've done is we've used inter-subject synchronization as a way to characterize the neural activity that kids have while viewing movies. And then we asked whether or not oxytocin receptor methylation is associated with more mature neural activation patterns. And I'll talk about what we mean by more mature neural activation patterns in this case. So in general, what we used was, we used inter-subject synchronization analysis. This really is a technique that was around early in 2000s. Yuri Hassan from Princeton studied a bunch of individuals looking at the movie Good, the Bad, the Ugly. And you can essentially take all of your subjects and you can find areas, you can quantify this at the voxel level, you can do different parcellations if you'd like. But in general, what you're looking for is voxels or whatever parcellation that shows a high degree of congruence across the time series in all of your subjects. And so in movies with really rich visual content, you can see this very easily in primary and secondary visual cortices that people generally have similar neural response across these systems. But they also do in some of these higher order cognitive systems that process the content, the emotion, the meaning of the stimuli as well. And essentially what you're doing when you're doing this analysis is you're decomposing the signal into sort of three things by looking at the relation for each subject to every other subject is you're looking for signals that are consistent across viewers, signals that are idiosyncratic, so might be real signal, but they are kind of unique to that person or unique to a group if you have multiple groups within your larger group of analysis. And that might be part of that signal. And then the third part is just spontaneous, and this could include noise, artifact, other things that we are accustomed to in the scanning environment. And what we did with this is we borrowed a technique from, well, let's see here. Let me just show you really quick this. This is the social emotional content of stimuli influences intersubject synchronization. So you could see this both in the children and adults, these three different conditions. The Inscapes condition, again, is those 90s screensaver looking thing where you have just sort of the real calm geometric shapes moving around. And you can see a lot of similar activity here in the primary visual cortex, a lot of motion sensitive areas, because there are things on the screen to process and there is content there, but it's more sensory content. There's nothing in terms of meaning. When you get into our NEMO clips, we had a clip that we call NEMO control, and we have a clip called NEMO social. The NEMO social clip is a really high arousing situation. It's for anyone that's seen the movie, it's when the fish killer Darla has Nemo in his bag and the other fish in the tank are trying to work together to save Nemo. And the Nemo control clip, it's just this more humorous, lighthearted, but really visually active sort of scene at the beginning of the film. And you can see these types of films have much more degree of synchronization across multiple areas of the cortex because the complexity of the content in them. Now, what we did was we borrowed a technique from Cantillon and colleagues, whereby they used these intersubject synchronization maps to then find differences between children and adults. And we could then find those differences and assess the degree to what we would then call neural maturation of these systems. How much does a kid look like the average of the adult map or the average of the adult ISC? And then we can quantify it. So you can see here in this pairwise ISC values within the adult children neural synchrony map. What we're going to do is these are our adults. So the participants are ordered by chronological age. So you just basically take all of your participants and line them up by age, and you have their time series from any given area. And then you have your participants ordered by chronological age coming 0 down to here as well. And so these folks down here in this square are 28 adults. And what we can do is for any subject, for any given parcellation, for any given voxel where you're looking at, you could establish the degree to which that participant is adult-like or child-like. So are they more, how congruent are they with our adult sample here versus not congruent with the adult sample? With the indication being if they're not congruent like the adults to the adult sample, they're more immature, developmentally immature in these systems. So we use this as this neural maturity metric as another way of assessing maturation of critical systems for social and emotional content. And what we find is the degree of oxytocin receptor methylation is curvilinear associated with neural maturity. So our hypothesis is that we would find higher levels of oxytocin receptor methylation being associated with more neural maturity, more adult-like profile. In this case, we actually have this more curvilinear association, which sort of begs the question is what's happening in some of these individuals that we have with really the highest levels of oxytocin receptor methylation. There's a couple of theories we have about that, but I don't want to go into that too much today, except for to say that there's an interesting pattern of data here. And then I want to unpack it a little further. You can see that, again, when we get into 2 years later, parent reported empathic behavior. So how emotionally mature is your child in terms of engaging in empathy with others. What we find is the kids that have lower levels of neural maturity and lower in oxytocin receptor methylation at the time of our test, of our MRI test, 2 years later, these are the kids that are more empathic. And I draw this circle here because I'm really interested in these children because a lot of the neuroimaging literature on kids and maturation of neural systems is focused on either deficit model or bad model, sort of bad upbringing, what's the consequence of bad environments on neural development, which obviously is an important question to ask. But one of the things that is frustrating from the other angle is that we don't really know what's good in terms of what makes an environment good and what are the consequences of that. Here, one of the suggestions and one of the hypotheses, and this is just the first data point in many that we're collecting and that I hope to over the years, is to show that some of this more protracted development might lead to more complex neural mechanisms that support larger, more pro-social types of behaviors, such as cooperation, empathy. Curiosity would be not pro-social, but would be another behavior that potentially support creativity, things that a more complex brain might support in this way. So to take home from here is that more adult-like neural synchrony during pro-social movie watching is curvilinear associated with oxytocin receptor methylation, and lower oxytocin receptor methylation and slower maturation is predictive of more empathic behaviors later in development. And so some of our future directions are one is that we're really trying to increase our sample. We'd like to start a new longitudinal cohort, have a little bit more variability in it, recruit more from the outlying areas to get out of Charlottesville a little to increase the diversity of the sample and try to track these things across time. We still don't have a great way from our data of understanding some social and parental variables in our preschool children. We do in our infant samples, but our preschool children, we have less ability to understand how that's driving some of the changes in oxytocin receptor methylation. So we want to do more work on that front, and that requires some more effort in terms of starting new samples or new data streams. And then also, in the second point, trying to understand in an era that we live in now where there's a lot of debate about early care, early preschool, government-supported preschool plans, things like that. What constitutes a good environment? Not necessarily to slow everyone down, but if there's an advantage to creating situations that provide more security and less need for self-regulatory process, does that allow other neural systems to develop in meaningful ways that support these other sort of societally sort of important behaviors, such as pro-social behaviors, such as empathy, compassion, cooperation, and things like that. And so that's where I think some of the direction of this research will take us in the future. Just briefly, I wanted to acknowledge my collaborators. As I said, this was work by Amalia Skyberg, who's now at the University of Oregon. She works there with in Jen Pfeiffer's lab, where she's doing studies in adolescent social development. And she continues to collaborate with us on some of these projects as well. And my longtime collaborator, Jessica Connolly. Ben Newman's the guy that does all the tractography studies, and the rest of the crew here is involved in a lot of the basics of putting together new analyses and some of the subject running and things like that. Thanks, and I'm happy to take questions.
Thank you for a very interesting talk, Dr. Morris. I would like to remind our audience once more to address any questions using Q&A box or raise their hands. Is there any questions? Yeah, then I have a question, Dr. Morris. So
okay,
it's more could be general kind of like curiosity about the measure of the oxytocin receptor methylation from the saliva.
Yeah.
So is it like well correlated with the brain tissues or kind of other kind of like gold standard measures?
Yeah, the degree to which we have been able to test this in humans and voles, there's a couple of disconnects here and there. In humans to test that, obviously we need postmortem tissue, which was established in the first study, and there's a high degree of correlation. In the voles, we don't collect their saliva through there. It's hard to assay through there, but in terms of other tissue types, blood, brain, urine, they find high correspondence between those tissues. So it doesn't really work like that very well in a lot of targets across the epigenome, but these areas of the oxytocin receptor promoter tend to show this sort of pattern across the various tissue types that we've tested. So it's made it an accessible and novel target to look at in this case.
Thank you. And to follow up the question, so could you remind me that what was the driving force to like, I don't know, it's operate to say that this way, but like the accelerate the prefrontal like modulation on amygdala on like children? Yeah.
I think the idea behind it, and it's hard to give single sort of explanation to complex biological processes. The idea is the uncertainty in environments with less sort of caretaker accessibility to manage difficult, stressful situations influences trajectories of neural development. And this is one of the systems that most is probably influenced the most by those environmental factors because it reduces stress overall. So if the organism's sort of has more of a central goal of stress reduction because of the dangers of it across the body, you might see these sort of adaptive mechanisms taking over to develop this circuit a little bit quicker. And I think that's where a lot of the early work sort of focused on why this was happening and why this was happening, particularly in children where there was less likely a caregiver accessible.
Thank you. And we have one question from Maria.
Yeah.
I wonder, is it possible to hear me now?
Yeah.
Excuse me?
Excuse me.
Oh, I can hear you.
Is it possible to hear me now? Yeah, thank you. Thank you very much for a wonderful talk, very inspiring. I'm just wondering about the reversibility of the findings that you're presenting. So I wonder, are there any studies or you're planning these studies, how fast actually the methylation can change if you change the environment? So like if a child is getting from a, let's say, institution to a foster family or something like that. And are there any therapeutical perspectives of the research that you're presenting?
That's a great question. It's not known at this time. There's a couple of angles that we're approaching this from. From my collaborators' angles, they're engaged in a lot of cross-fostering studies in terms of the prairie voles in the lab. So you can take, pups that are born of low care parents and put them with high care parents instead to see how that manipulates it. That doesn't specifically address your change because now that trajectory is more linear, just changed at the beginning. But it does sort of get to this argument of, whether or not there's something else driving this or what role the caretaking actually does have in terms of changing this over time. In terms of change in the humans or in terms of measuring it, in the humans, it would be a little bit harder to speculate. We're trying to get an idea of when exactly we see the biggest change, whether it's in infancy or early childhood, and that requires lots and lots of subjects, not necessarily ones we scan, but just a lot of tissue from there. And then also, we've been kind of keeping our eye on some of these studies where people have been developing more of these epigenetic whole genome clocks, which also capture a lot of this similar variability. And some of the more new age, new era clocks are more sensitive to this ability to collect and change and measure over longitudinal time. I'm interested in that because I think that this would specifically speak to what you're saying, which is that it's possible that some specific changes are made or some specific events are what's causing some of this, and then that event is removed. And then you might see a slower rate of change afterwards, or you might see some things that accelerate the rate of change. And I think that those are really fascinating questions, and they're ones that just, unfortunately, probably take some years to track, or some very specific sort of insights from the model systems work that we can then find some populations where they might have already measured these things and might be able to look at tissue from there, because doing it as a single lab would take a lot of efforts to be able to fully unpack all the various angles that you would have to come from to answer that.
Thank you. Yeah, I have several other questions. I don't know if we have, do we have anything from?
No, you can go. You can go.
Yeah, just it's really fascinating that you're presenting this link with the connectivity of the brain and this probably compensatory mechanism that you're showing with this early maturation. I just wonder, do you look at the same time on other systems which are underdeveloped, and how tightly this link is? So if you have a higher maturation of the emotional regulation, does it correlate whether slow motor development or speech development, etc., that we see in this kind of, like, I'm just thinking about the kids in unfavorable conditions, let's say in an institution.
Yeah. Again, it's sort of one of the things that I'm really sort of interested in looking at myself and hoping to get more data on, because there's a couple of questions that come with that. One is that if I just took our own longitudinal sample, I was just looking at different outcomes, I would be led to this idea that higher levels of oxytocin receptor methylation are associated with more mature neural systems, and in many cases, more favorable behavioral outcomes. These kids have better metrics of self-control. We actually see some better metrics of various cognitive tests as well. We test them on things like flanker test performance and things like that. And so if I was just taking that straight from what the data say, I might be led to say, hey, kids that have higher levels of oxytocin receptor methylation do better. And then that kind of leads into this case of like, okay, but you said before higher levels of oxytocin receptor methylation might be more associated with lower care contact parents, things like that from both the animal and human studies. This can't be right, which either could suggest one of two things. One is that maybe there's more of a sweet spot of, and we're just capturing that these kids that we're calling lower care are just kids that aren't being helicoptered. Or maybe it's that we haven't really realized the full ramifications yet. In that early on in development, this might look favorable, but later on in development, this might lead to sort of more other deleterious or less favorable outcomes. And we just haven't seen the whole lifespan for that yet. The other side of that, which is what I said with our kids that are lower in oxytocin receptor methylation and neural maturity, I did point out that 2 years later, they show a higher amount of empathic behaviors. So maybe there's some of this is coming on board. And we do have this interesting that I didn't report here because it's kind of only preliminary, but it looks like if we look at some of the white matter tractography, they have more complex tractography in terms of longer range connectivity than our kids with lower levels or higher levels of oxytocin receptor methylation. So I'm, interested if I had a bigger sample of doing more sort of through functional connectivity, doing more network dynamics to establish whether or not we can get at some of the levels of, are there complexity differences in the ways that the brains have developed, which are then going to support more complex behaviors as the kids get older, which these kids that are slower developing might actually then be advantaged later, but not seeing that advantage when we're looking at them when they're 8, 9, 10 years old. And that's where I really think that there's, I don't think we know the answer. And I think that that's an interesting question.
Thank you. Yeah. Okay, if we just very short follow-up thing. If you look at some of them just for the basic measurements of the stress, like cortisol, so like, do you think that in these kids, who have this, early maturation, they have actually, let's say, higher cortisol? Is it known? Because this is something like a very simple measure to
yeah, we don't have that yet, and we do have. Again, it's like we all had a little bit of an increase through the stress response there. I think in general, part of the problem we have is that we still are only looking at one end of the spectrum. And, the variability we are capturing is meaningful, but it's not as meaningful if we had kids that had more significant early life history, early life events. And it's a case where we'd have to be more expansive in our sort of catchment in our population. And that's what we're trying to do. In our own studies, we're trying to engage more outside community, go into more of the outside communities, both within and outside of Charlottesville, to engage more families to see if we can, increase diversity, so we can better understand this. But yeah, I think we should take more of those measures, and I think we would. And I think the first step to that, though, is to get more representational in our sample.
Thank you very much. Very fascinating. Thank you.
You're welcome.
Okay, and I have one more question. So
Sure.
was there any, I know like the like epigenetics not necessarily like inherited, but like was there any correlation between the oxytocin receptor methylation level between the kids and like the parents?
Yeah, I think there's a moderate level of association between them, and we don't know the nature of it, if it's sort of environmentally impacted and genetically linked, or there's obviously a lot of different theories that get more provocative than that, but that again is something that we can get a little bit more of through some of these generational studies in the voles. In terms of the human studies themselves, we need much larger sort of just a sample of available tissues and work is being sort of done on that, but we haven't got as far with it. So yeah, I think that's an important question. And the source of this, really, the mechanisms by which this is actually occurring are under a lot of study from the model systems work, from early pregnancy studies and things like that. But we're only halfway around to unpacking some of those bigger questions about the source of the methylation.
Thank you. I'm actually
Yeah.
like impressed by your works as a like father of
Thanks.
2-year-old son.
Yeah, it always comes up. The parents, like, especially when we do the vole talks, because the vole talks really get into the parenting, and everyone's like, "Oh my God, I got to go and think about my kids."
Again, thank you very much for giving us great talk, Dr. James Morris.
Thanks.
And thank you
Appreciate
everyone
it.
for joining us
Yep.
today.
All right. See you soon. Thanks a lot.
Thank you.